If anybody has questions in the audience, Logan.
Thank you. This is obviously amazing and has clear therapeutic potential. It would also be great for the scientific neuroscience community to access some of these tools. Do you have any plans to make these available to neuroscientists?
Yes, we do. It's a great question. I think there's probably a lot that can be figured out if we provide the surgical robot and devices to neuroscience research departments at universities and hospitals. So I think at the point at which we need to be in production with the machines and obviously have the FDA approvals, but I think it would make a lot of sense to provide this to research universities and hospitals. The question is, of the data that we...
Of the data sets that you've collected, are there any that you plan to open source for the scientific community?
Yeah, I think that would be fine. I think, yeah, sure, absolutely.
Because I think it could be really interesting for people working in AI research to build upon that and build foundation models for the brain.
Yeah, it's a good point. Yeah, I actually got no problem with just publishing it on our website. You can use it if you want.
Yeah, looking forward.
Great.
You go.
Thank you. Thank you for the very wonderful presentation. So I have one question. So as we know, for implantable electrode, either for stimulation or recording, after we implant the electrode, the scar tissue will grow around the electrode, and especially for recording, the signal we get will become smaller and smaller after long-term implant. How do you solve this issue?
For context, I'm Zach. I lead the microfabrication team on brain interfaces. I don't think we can solve it specifically, but one advantage we have is both the flexibility and the small size of our threads to try to limit that scar tissue and that damage. And some future work that we have started working on, that we'll continue working on, is pushing the size of the threads down, just to try to limit the immune response and really limit that scar tissue growth.
Actually, I want to follow up. So, do you think it will be helpful to actually load some drug on the surface of your electrode, or some other way?
Well, I think like maybe the question is like what sort of signal degradation have we seen over time, and basically does it work a year later? Does it work 2 years later? It does. So, yeah.
Yeah. So that's a good point. So. In terms of thread longevity specifically, really the gold standard that we can use to assess is the data we have from our animal participants. And so for that, I'm not sure if it was mentioned before, but the longest data we have right now is for an animal participant who went 600 days with useful functioning channels where we were doing something useful with the signals for BCI. And then with the newest version of our device, we have sort of a collection of participants who are at or near 1 year of data and completely useful functioning BCI from that as well.
Thank you.
If I may add one more thing. So you mentioned potentially having drugs to kind of reduce inflammation. So one of the things that we are actually actively working on is having some sort of biological coating to either reduce inflammation or make them slippery. So you mentioned, you heard from the presentation that one of the challenges that we have is removing the threads from these neomembrane tissue that are formed after implantation. So there are programs like that where we're really looking at kind of incorporating some of the learnings from biology and these coatings into our threads so that we can hopefully reduce inflammation as well as make it easier to extract.
We're also continuing to reduce the size of the electrode. So when the electrode gets really small, there's the sort of inflammation response or scar tissue becomes minuscule. So it's like a very tiny electrode the body basically ignores.
This is really impressive. Congrats to the whole team. So as you of course know, one of the problems with current electrodes is they're rigid and they move around, so you have these neural non-stationarities. And I think many of us had hoped that with these very thin threads, they would maybe move more with the brain and you wouldn't see that. But from the data you showed over many hundreds of days, there was a lot of variability. So can you speak to how much do they move, and do you have any idea of like why does it move? Can you stop it from moving? Basically, how stable are the signals hour to hour and day to day?
Hi, I'm Bliss. I'm one of the leads of the software groups in the brain interfaces team. In the particular plot you were mentioning before, what we were showing was the average firing rate recorded per day on a particular channel. It's, as you well know, pretty complicated to understand if you're recording from the exact same neuron day after day after day. It could be, for example, that you're actually picking up a different neuron day to day, and that's why you get the change in firing rate. We don't think this is at least the majority cause of the situation here. The reason is that if you look at sort of the spike shapes day to day, even when the average firing rate is shifting a lot, you still see sort of stable spike shapes. That's obviously not a fully bulletproof story, but at least gives some confidence that it's not actually different neurons you're picking up. However, there's very much a chance that that could be the case in at least some part of the robustness non-stationarity story. Yeah.
Cool, thanks.
Yep, thanks for the question.
Yeah, to be clear, that like this electrode position is actually fairly stable, because you've got these very tiny, basically very tiny wires with there's some play in like you've got the device attached to the skull rigidly, but then you've got this long sort of tiny wire with kind of a coiled section. So it does tend to basically stay in the same place. Yep.
We also asked for questions on Neuralink's Twitter, so we'll be interleaving some of those. Supe wants to ask, what could Neuralink help people with that most people don't realize?
Well, I mean, so once you're in there, there's a lot you could do. You can obviously measure temperature, so you could do very early detection of a fever. You can measure pressure. I think you probably detect that at the very beginnings of a stroke, because you can see sort of like electrical signals starting to go sort of haywire. So there's actually probably a lot of just general health monitoring that you could do once you're in there, and with very simple sensors.
Hi. You guys all did a great job of distilling a lot of complex engineering and science and making it wonderfully clear, so great job. I wanted to ask a little bit about the stimulation, I guess, for the phosphenes and for the evoked movement. How are you thinking, is this more like local stimulation? Is it juxtacellular? Are you steering current around? How many cells are you activating? How much current are you using? I'm just curious what the scale of this is and whether you have a lot of precision or a lot of, you have pretty profound behavioral effects too.
Hi, yeah, I'm Dan, and how many cells you stimulate with a single electrode is dependent on the impedance of the electrode, size of the conductive pad, how much current you deliver, the frequency, all these factors. So there's a great deal of variability that we can use to customize the shape of a phosphene, or not the shape necessarily, but maybe the intensity of a phosphene. We think with our current electrodes, at least in code, back of the envelope calculation would be something like about a 50 to 100 micron diameter sphere of cells are being stimulated. In a visual system, the smaller that sphere, the smaller and more specific you can make a particular phosphene. Basically, the smaller the pixel in the image you can produce. So there's plenty of scope for customization of that.
There's actually also, it's possible to get to a much higher effective pixel count by controlling the field, electric field, between the electrodes. So it's not necessarily, it's not a one-to-one relationship. You could actually dynamically adjust the field and simulate have a very high neuron to electrode ratio. So try to could you get maybe 10 to 1, 100 to 1 potentially. So a megapixel type. I mean, basically, can you see normally? I think people would want to know that. And I think that is one of the possible outcomes.
Hi, Elon. This is amazing. Can you talk about the longevity of the implant itself? Also, how would the material of the implant would react with the brain tissue or density of the bone or bone structure? Thank you.
Yeah, happy to talk about this. I'm Jeremy, an engineer on the brain interfaces team, and I think it's good to start with data. So, like Zach mentioned, we have an implant that was, a monkey was performing BCI for 617 days. That was pager before being upgraded to the latest device. For our current version of the device, it's lasted for almost a year. And then for our accelerated lifetime tester that Josh kind of talked about, we have data from our implants from the previous version, 8 years of accelerated time, and from the current version, 4 years of accelerated time and counting. So that's kind of starting with the data. Those devices are still lasting and still going. Theoretically, there are kind of 3 fundamental factors that contribute to the longevity of the device. One is going to be the seal, the hermetic enclosure of the device. 2 is going to be the battery and internal electronics. And then 3 is going to be the threads that Zach talked about a little bit. And the channels being able to functionally record signals from the brain. The seal, we think, will far outlast the other 2 in terms of the bottlenecks. So the seal, just theoretically, I think Josh mentioned that it is a thermoplastic polymer material. So there's going to be a very small amount of moisture that diffuses through it over time. And we think that will last 20 plus years easily in terms of just that property. And like I said, we have not seen our seals fail with our current version of the device yet. So we haven't really pushed the limits here. For the battery and internal electronics, that's really based on usage and how much runtime you want. And we are working currently on getting data to project out even farther. But right now, we believe that we can achieve 80% runtime at the 3-year time point, which would be about 3.5 hours for a 4-hour runtime. But we're, like Avinash mentioned, we're doubling that very soon and quadrupling as well. We have plans to do that. So the internal electronics really aren't the bottleneck either. And so really we're attacking the threads themselves and the longevity of those channels that Zach can kind of talk about some of the improvements that we're doing to increase that longevity.
Thanks. Yeah, so as sort of mentioned before, we don't necessarily have an endpoint, as Jeremy said, for the testing of the threads. That being said, we are focusing on longevity because we think this is an important issue to solve. So one thing that we're doing in parallel with the current device is aggressively pursuing amorphous silicon carbide insulation of the threads, which we believe will take us well beyond 5 years of longevity, but of course still to be tested. And in parallel with that, we're just starting to look at atomic layer deposition, which we think could even push longevity of the threads much further, and deposit very thin layers to keep the flexibility of the threads and that advantage there. Along with that, we're also, of course, having to design and validate very robust benchtop testing to model really in vivo conditions and look at channel degradation. So that's what we're looking at for longevity of the threads. And then I think you asked about biocomp. And I think for biocomp, essentially all the materials we're using right now, I can say are at least biostable, and we send out testing for biocompatibility very often. And essentially what we're doing is we're using, in many cases, known materials from literature that academic labs have already started to look at and sort of jumping on that and using that as a starting point.
Thanks for answering that.
Cool. So we have another question from Twitter. This is from David, and he asked the team, what are the biggest lessons you learned since the previous presentation? It's been about 2 years. I'm sure there was a lot of engineering done. So yeah, anyone want to answer what we learned in the last 2 years?
So one thing that we've learned in the last couple of years is just how much the brain moves on the human scale compared to, when you start small, when you make brain proxies, and a lot of research starts with rodents. The brain does not move that much. And when you get a human, and the brain can move like hundreds of microns or more. And when our threads and needles are so small, that motion, when you zoom in, looks like a mile.
I think to add to that, one thing is how, I guess, dynamic
the implant environment actually is. So we've talked about like when the implant site heals, scar or new tissue might grow and fill in the space, and that'll affect like how our threads might interact in that space. So that's why we've emphasized so heavily the importance of designing accurate proxies. So instead of having to wait months for an implant site to heal, you can hopefully learn that information in hours.
I'm Alex on the robotics team. I think one of the things we've definitely learned within the engineering teams is the importance of really continuous validation and testing, where we're building, say, motion systems that are precise to single-digit microns. We need validation and test systems that we trust even more than that to prove that they work reliably. And putting just as much focus into those validation and test systems and designing those alongside our products, I think is one thing we've definitely learned.
I think another thing that we learned, I think, as part of BCI or the brain control and algorithm, is that, again, building a prototype and making it work with only one monkey, one pager, was a great, maybe a success, but also relatively easy to making it work every day for all the other monkeys. So actually making it a product is something that it's not easy, but we are learning how to do it.
I mean, I've learned that the brain is really squishy. Like way squishier than you think. It's not like, cauliflower or broccoli or something like that. It's more like a water balloon. And then it's moving in your skull like a lot. So you got a squishy water balloon in a coconut. Is maybe a good way to think of it.
Hello. Given Bluetooth's bandwidth limitations, have you considered other technologies for wireless communication?
Hey, yeah, I can take the first part of this question, and then I'll let Matt answer the second part of it. It's a great question, especially as you think about how to increase and scale the number of channels that we want to record from. This becomes increasingly a bottleneck for the kinds of work that we want to do. We're thinking about this in a couple ways. One is just directly improving the underlying radio interfaces, and I'll let Matt talk about that in a second. The other way we're thinking about this is how can you be more efficient with the data you send off the implant. And I think the first version of that is compression. So just taking your data, looking at the characteristics of it, find out a way to represent it more efficiently and just send off that compressed stream. So for reference, right now, our Bluetooth bandwidth is around 150 kilobytes per second. The compressed stream of data that we send off the implant is around 50 kilobytes per second. So we're doing fairly well there so far. But when you start thinking about 16,000 channel devices, that won't get you all the way there. So some other things that can help on the compression side are to actually just send out the output of the machine learning model rather than the input required to actually run it. So one thing we've been trying in the background here is called decode on head, which is essentially taking the machine learning models that right now we're running on MacBooks that our monkeys are gaming on and moving those to actually run on the implant. And this is like a super cool engineering problem. If you want to talk about how to make complex neural networks run on what is the equivalent of a garage door opener, come talk to me. It's fun. Yeah, so that's another way to solve this problem, is to basically do the computationally intensive work to just get the raw signal that you actually care to use to control something, and then send that thing out of the implant. On the radio side, I'll hand it over to Matt.
Yes, so to answer your question, we are looking at other radio technologies. One in particular is 500 megahertz band with ultra-wideband at a couple different frequencies. So this has an advantage in terms of the bit rate that you can achieve. It's on the order of 6 to 8 to 10 megabit. There's also a latency improvement that's quite substantial. And there's also another wireless technology that we're looking at W-band.
Hi, thank you all for really clear and compelling presentations. Something that struck me in one of the earliest talks, I think it might have been DJ's, was this vision for the ability to acquire new complex skills via these BCIs, like the ability to perform Kung Fu. And that reflects the fact that the brain is fundamentally a learning machine. And yet, many of the technical solutions presented later framed were framed in such a way as to try to correct for the way the brain changes over time, over longer time scales, drift over the course of days, or the way that the tissue might heal over time. I was curious what your vision collectively was for developing out this technology that interfaces with a fundamentally plastic system that changes in complex ways over a variety of timescales, days, months, years.
Yeah, it's a tough question. Yeah, I think it will be kind of maybe bidirectional learning in some way. In there are sometimes scales that might fix our algorithms, and we prefer to have like more stable kind of performance. But of course, if over time the person and the brain will learn how to use better the BCI, we'll need to update our models. So there will be kind of an interactive kind of relationship in some way. To learn even new tasks, this probably will be something over time we'll need to learn what the person kind of learn how to interact with the computer, and then build the appropriate interface, the UX, and also the UI, and build algorithms that will help him to control what we want.
Just one thing I'd like to add on to what Nir said. Yeah, it actually is an advantage in some ways that the brain is plastic and learns, and that can help us because we actually have to do less work, and the human in the loop will actually learn how to use our device better. But one of the advantages of our particular approach and device is that we are trying to do an extremely high channel count device, so we can uniformly distribute electrodes over a functional region, and then it doesn't matter so much whether things move or shift over time. We can offload that to software. And so we can build algorithms that change over time as well. And so both those things are actually, I think, advantages to our particular approach.
We have another question from Twitter. Juan wants to know, what career path do you suggest for somebody that is just getting out of high school if they want to work at Neuralink in the future?
Well, it's really any of the skills that we described. We're developing new chips, there's material science, there's software, obviously, animal care. I mean, it's really all the things that we listed in the Neuralink /careers. That'd be a good guide.
Yeah, I'm actually very fond of saying, when you flip through any college booklets and look through all the majors, I think you can point to every single one of those majors, and there's someone at this company who either is an expert or have majored in that. So it really is truly multidisciplinary endeavor. And I think just focus on whatever you're passionate about or whatever you're talented at, and then just pursue that as deeply as you can, and then there's definitely going to be a place for you in building neural interfaces.
Hey. We got to see the monkeys doing telepathy, but could you say a little bit more about the animal behavioral training, kind of their lives and day-to-day processes?
Sure. I'm Autumn. I am head of research services, which includes our animal care program. And as an animal welfare scientist, this is a topic that I'm deeply interested in. So our training program is outfitted mostly with behavior analysts who help us think about how to remove any of the potential aversives or frustrations from our training. We think about conditioning as the primary, which includes positive reinforcement, as the primary way to train. Let's see, what else can I share with you? Yeah, I mean, that may not be part of the behavioral training itself, but we think of animal welfare assessment in the framework of the 3 Rs, which is refinement, replacement, and reduction. And so when we think about refinement, behavioral training does apply in that way, and where we want to remove, specifically in research, restraint is one of the things we make a very top goal to remove. So you saw a lot of videos today where animals were walking up to their stations because we worked really hard to remove any requirement to restrain the animal. Anything else?
Yeah, if I
to add
can
to that?
just,
Yeah.
well, just on top of the last point you said, just as an engineer here, one of the things that is really inspiring and really cool about this place is that we do get to work on a lot of technological innovations that directly translate to greater independence for the animals when they're engaging in these tasks. So as you saw, monkeys charge just by voluntarily walking up to a branch. They play games in their home habitat with a laptop computer voluntarily. And the fully implantable, fully wireless device, the inductive charger, all these things enable that kind of experience. And so this is one of the very cool parts about working here is we do get to innovate on things like that.
Definitely helps to work with a group of engineers who can really make cool stuff for monkeys to be able to do easier behavioral training.
Yeah, so I guess to answer the previous question about what you can study to be part of Neuralink, I guess monkey engineering you can add to that.
Monkey business. Hello. Oh, it's on. My question is on upgradability, which you guys mentioned quite a bit. So in that procedure, I imagine there's some kind of explant procedure, and then you're going to put in a new set of implants. So could you talk about the damage possible, if any, tissue damage from the explant procedure? How long you have to wait? Do you implant in the same areas? And what's your brain scanning for the implant procedure in terms of upgrading it? I don't know how many questions I can ask, but I have one more after this too.
So I can start to speak to some of those. So I work a lot on upgradability and those explant processes and designing those to be better. The goal that we're working towards is that, as I mentioned in the presentation, it's really just as easy to upgrade an implant as it is to initially install. We didn't show many of those explant examples today, but we've come pretty close to just popping out an implant and reinstalling another one in the exact same location. Definitely the goal, we are installing the implant in primary motor cortex, which is a valuable area for interacting with a device like this. And so the goal is to implant in the same location. Maybe if you expand out to other applications, then you'd be interested in moving somewhere else. But we definitely want to be able to insert into the same area. In terms of damage, I think that the damage that we care most about is damage within the brain. And what we've found, and we talked about that challenge of the tissue layer on top of the brain, and I think we're well on our way towards figuring that out. But because of the thread's small size, the sort of scar capsule within the brain is so minimal that they are actually removed quite easily. And so we see useful signals even on the second or third time that you've placed an implant. And I think some of our BCI folks can probably speak to that. We do have monkey participants working with their second devices and really making use of those.
So two questions. One was somebody had asked the question about the brain being plastic. Have you noticed any plasticity from a behavior perspective from any of the monkeys? Or is it too soon to tell, or there haven't been any observations?
From the monkey behavior, we see that it takes them a while to learn how to, of course, to train on the task, but also when they are reimplanted. It's relatively quickly for them to ramp up and get to a high performance of brain control. With Pager, for example, after a few days, he was able to, 3 days already, able to learn very quickly to use the device. He was trained on the task, of course, from his previous implant, but with the new one, after 3-4 days, he was able to control to close to performance of he had with the previous implant.
But have you noticed anything on the adverse side, which means the brain has outpaced the neural network that you're running?
It's hard to say.
No, not really.
Okay. So I have another question, which is more about the electrical side. So you talked about 1024 channels being recording. Are you transmitting the raw signal, or was it only the 3 spike events that you were talking about, the low, mid, and the high? Or is it the entire raw waveform that you transmit?
Yeah, hi, I'm Julian. I can speak a bit about this, and maybe Avinash wants to contribute. But our chips see the raw signals, but then when we transmit out typically spikes, and we detect those spikes in real time on the chip, this massively compresses the data. I guess, yeah, we're making improvements to that, but we can request raw samples. Sometimes we also process particular statistics or other data directly on the chip and then send out the calculated values. So there are many ways to sort of play with the data.
Yeah, so at least with the current N1 system that we have, which relies on BLE radio, there is a bandwidth limitation, so you can't actually stream raw data from all 1,024 channels. But just kind of to give you a little bit of a history of how our compression algorithm, the spike detection algorithm, was developed. We did have sort of a wire system. There was a paper that we published with the USB-C connector that streams all those signals through a high bandwidth wire connection. So we did have kind of those development platform to be able to see the raw signals and know which set of information that we want to extract that are going to fit within the bandwidth of the radio, as well as is useful for BCI control. And also just sending data wirelessly does cost a lot of energy. So there's any opportunities we have to reduce that burden. We try to basically have all that compression closer to where the electrodes are as possible.
Maybe one thing that isn't obvious is that the actual bit rate that you need to control a phone or a computer is actually very, very low. So I think we might have the record for bit rate. Is that correct? We think we do, maybe. So on the order of 10 bits per second. So that's super slow. But if you think like when you're inputting data into a phone, like how fast are your thumbs moving? What's your thumb taps per second? Pretty low. And we're like basically our thumbs are like 2 slow-moving meat sticks that we, do this, and it's like it's really low. It's like a low bar, is what I'm saying. So at least for output, it's you get 10 bits per second. You're hauling ass. And that's you don't need Bluetooth anything for you. So you could practically send it out with beeps and bops. So it's not. If you're going like high bandwidth visual. Now you're maybe going to megabit plus, but it's still really well within Bluetooth. But anyway, it's just that is what I'm saying is that's not a constraint, the data rate. One of the sort of like maybe notable item which we talked about in the presentation, but we think we can probably solve for doing the implant without cutting the dura. We can just do basically a bunch of holes through the dura, which is like the dura is like the big thick orange rindy thing that contains that's up against the skull. If you don't pierce the dura, if you don't cut the dura away, and instead you have a bunch of tiny holes and insert the electrodes through the tiny holes into the brain, then the recovery time is ridiculously fast. You're not really losing much in the way of cerebrospinal fluid. You could in theory, this could be like the whole thing could be a 10-minute operation, like LASIK. Like it's fast. It's not like a big laborious thing. It's super fast.
Just going back to the long-term use, I'm wondering if you have any pathology looking at scar tissue from any animals that have had long-term implants. And along that lines, it seems like there might be a little bit of a gap between use in medical conditions and healthy individuals from a safety perspective.
I didn't quite catch the last question, but I heard the first one. I'll ask you to repeat the second one. So the first one is, do we have pathology from long-term use animals? We absolutely do. We don't have any pathology from our monkeys, which we upgrade and are still going. We have other studies that are primarily to determine safety, and so we do have histopathologic endpoints that we determine. The scar tissue formation around the threads themselves in the brain is typically negligible, like it barely reacts to the threads at all. So that's very promising. In terms of the scar tissue formation over the cortex, so this neomembrane growth that fills in the areas that Elon and Alex were mentioning that we remove with our current operation, those we do evaluate that scar tissue, but it doesn't pose a problem in any way. It's not a continuous reaction to a foreign body. It's just filling in tissue that was removed. And if you could repeat the second question, I didn't hear that.
Yeah, the second question, really following up on that, seems like there might be a little bit of a gap in use in healthy individuals from a safety perspective. I think people mentioned that they might be interested in trying prototypes, but just wondering what your perspective is on trying to lower the safety risks.
Yeah, it's a great question. So in terms of, really it's about the long-term use of the device. So we have devices that have been implanted, like I said, in monkeys where, for many years where we see no behavioral deficit at all. So this first is a question of how you evaluate safety. So you have histopathologic endpoints you can evaluate, but we're also looking for cognitive deficits or behavioral deficits as well. And we don't see any of those in our animals, which is an important point. In terms of the histopathologic endpoints, they look really, really great. The challenge is one of explanting the device, which is why we're putting so much effort into the reversibility efforts and our through dura insertions. So when removing the device, that's when you potentially could cause damage. And so we have a lot of ongoing studies right now to really minimize the risk of that. But we don't think it's a substantial risk with our current approach. And like I said, Pager was upgraded with the previous surgical approach and is doing great. So clearly it is, can be perfectly safe, but proving that beyond a shadow of a doubt for humans is something that we're still working to do rigorously. Did that answer your question?
Yeah, thank you.
So thank you for a very deep dive on many of the different aspects of the device and the system. It's very impressive to see all of the engineering work that's gone into it. You just mentioned about bitrate. As the prior bitrate holder, I can confirm you have indeed shattered my record. So congratulations on, I think I saw a peak of 7.4 bits per second. Well done. My question is actually around clinical trials and the FDA. To the extent that you can share, I gather that device removal or maybe electrode removal is one of the concerns that the FDA highlighted. Is there anything else you can tell us about what the FDA was concerned about or had questions about with respect to their IDE submission?
Yeah, I mean, we can probably talk a little bit. I mean, these are really challenges that we have broadly. So explantation safety, proving that rigorously for humans is something that definitely is one challenge and was something that the FDA commented on. Other things, they do ask some really great questions. So other things involve things like the thermal benchtop testing of our implant. So obviously it's important that our implant doesn't damage the tissue by overheating. So having really rigorous and valid benchtop testing for that is very important. It's actually something that we're redesigned to be even more accurate now. It's also the case that they ask a lot of very hard questions on biocompatibility, chemical characterization. So we've done very rigorous testing for that, but they do ask a lot of questions about getting into the weeds of the data and making sure that there really is no chance for any toxic chemicals or bioincompatible materials to be in the brain. So these are all things we're working with to just prove, again, above and beyond a shadow of a doubt. One thing that's maybe worth mentioning here is that it can be difficult to appreciate the novelty of our product. So the surgical robot and the thin film array in particular are quite new and unlike existing devices, and this means that we can't rely heavily on literature to support the safety and efficacy of the device. So we do spend a ton of effort in designing and performing testing on our devices so that we can rigorously prove the safety of them, and we can't rely just on another product or on some paper. And that's something that we're not willing to compromise for our first human participant and working very hard to do.
I think if you ask a question like in my opinion, would I be comfortable implanting this in someone, one of my kids or something like that? If at this point, if they're in a serious, let's say if they broke their neck, would I feel comfortable right now doing it? I would. I would say we're at the point where, at least in my opinion, it would not be dangerous.
Hi, thank you for the presentation. So I have a non-technical question. Are you collaborating with people with motor disabilities? And if so, like, have they shared any ideas of applications that they would be excited about?
I can take the first part of this. I'm not the best person to speak to this, to be honest, but there is a consumer advisory board we have made up of a number of people that have various conditions, including tetraplegia, and they give advice to us on a number of topics. Just as an anecdote, someone came to the office maybe 6 months ago, and they were telling me what they most wanted to do with their Neuralink device, and there were two things that they said. One was they wanted to be able to trade stocks day to day, to be able to beat their brother. And the second one was they wanted to be able to play shooter games. So I think what was most shocking to me about that encounter was the normalcy of that. And I found that conversation truly inspiring. So you know who you are, the person who came and talked to me. Have a great day. Yeah.
You know, something that's we've talked about, but is maybe it should be reemphasized. We are doing we're building up a production system for the devices. So we're bringing out the production line, making large numbers of devices. We want to make thousands, ultimately tens of thousands, then millions of devices. So the progress at first, particularly as it applies to humans, will seem perhaps agonizingly slow, but we're doing all of the things necessary to bring it to scale in parallel. So in theory, it should progress should be exponential.
Okay, so thank you. That was a very cool presentation. So one of the stated goals was recording from everywhere in the brain, being able to record from and perturb any location. So it seems like currently it's all cortical. And I'm curious, with the current device, is there any sort of long-term goal or idea as to extending it into going deeper in the brain? I mean, for neuropsychiatric disorders, for memory, all of these things are much deeper, several centimeters. So I'm wondering, what's the time scale, if you were to give a very rough estimate, of when I can expect to see a Neuralink product that goes that deep?
Yeah, so, I mean, the fundamentals of the device in the skull will stay essentially the same because as I said earlier, the device in the skull is very much like a smartwatch. Essentially, it's a battery, radio, inductive charger, computer, and then you've got the little wires. And so you need to make the wires longer, and you'd have to have a deeper insertion needle for the robot, but this really is intended to be a generalized I/O device. So apart from the tiny wires being longer and the surgical robot needing a longer needle, in theory, you should be able to go anywhere.
Because it seems to me that part of the robot is trying to detect where the blood vessels are and then avoid them, correct? Would that be possible at that scale? I mean, certainly not just visually, but maybe there's some other way of detecting it. Is that a current goal, and do you expect that within, I suppose, the next decade?
Definitely, yes. I'm Ian. I run the robotics and surgery engineering team here. Of the three axes that DJ mentioned, one of them is, access to more areas of the brain. So the robot team thinks about this a ton in terms of what sensors do you need to essentially go past the surface. And so in this case, you're right that right now we can really only see down the maximum about 1 millimeter. I think within the team, there's questions of what's best to use next, but like ultrasound and photoacoustic tomography are two that come to mind as things that can get centimeters deep, essentially. But it's a super interesting problem. You sort of need deep imaging and some ability to steer to at least avoid large vasculature deep down. Yeah.
Or if we can make our needles and threads small enough in a way that we can still be precise and accurate at a deep depth, then maybe you don't cause a bleed if you hit a vessel.
Yeah, I think that's really the ideal situation. If the threads are really tiny, they can actually go through a blood vessel, and it's okay if they're tiny enough. So we wouldn't need the blood vessel imaging in that case. I actually am slightly optimistic that is achievable.
And Matt, you could probably speak more into this, but with DBS currently, it's kind of just like send it.
Yeah, the current approach involves a wire that you blindly pass in that's massive compared to our threads, orders of magnitude bigger. And so that's a low bar for us to clear as well.
I guess people don't realize, like, for the deep brain simulation, just how big the hole is. It's what is it, like? I mean, how basically, in current deep brain simulation, how much of a borehole is drilled in the brain?
Yeah, you're drilling a 14 millimeter burr hole and then passing a 2 millimeter wire, 6 centimeters, 8 centimeters deep into the brain. So all blindly, hoping that you don't hit a blood vessel, telling the patient up front, this might be good for you, and there's a 1% chance your brain is going to bleed in a way we can't control. So
that is current technology that is happening right now. So doing better than that is we can definitely do way better than that. No problem.
Our needle's 40 microns in diameter.
Yeah, exactly.
Thanks again for the phenomenal presentation. I thought it was fascinating how rapidly you could test all of these electrodes, but it begs the question about like what your fault tolerance is. If you run these diagnostics and it comes back that you have something that's either shorted or high Z, how many of those before you get degraded performance? And the second question is when you're actually inserting this device, we saw examples of the electrode going in and then like looping back on itself, but it looked like that was something that was assessed basically by slicing the synthetic material. I'm curious what you're doing to validate the insertion of all these electrodes sort of in vivo. How do we know that that's not happening on an actual patient?
Yeah, I can answer that the second one. So like I mentioned, we can, so we weren't actually sectioning in that case. We have a really cool micro CT. It's essentially like a CT scanner. So that's just an intact proxy that we put in this machine, and we can take a picture all the way through it. And like I mentioned before, like we can make a proxy where it happens, that looping back happens every single time, and then we can make one where it never happens. And we've pinpointed roughly now where actual tissue falls in there. And so our current plan for validating and confirming that is making proxies where it happens really easily, much worse case than any tissue could possibly be, and then designing it such that it never happens in that scenario. And that'll give us doing that enough times and with a weak enough proxy, that'll give us the confidence that this isn't actually happening. Yeah, and this is the next gen needle. We don't see this problem at all with the current generation.
And I'll take a stab at your first question. So to clarify, you're asking what happens if there's a fault on a particular channel or something?
Yeah, that's correct.
Yeah. So the nominal scenario is that basically the impedance will stabilize pretty quickly within the brain, and even at that level we can record great signals, we see lots of spikes, and we can use that for BCI. Because we have so many channels, like 1,000 now, 16,000 later, we can actually run our models with far less channels than we actually have. So it doesn't matter if like one channel dies here or there, like we can still do really good decode. I'm not sure if we have official numbers on how many channels we need, but it's like we have an order of magnitude more, and the more we have, the better, but we can already do a lot with what we have.
Maybe just one or two more questions.
Yeah, I have a question about your very, very long-term inspiration to have this high bandwidth communication with advanced AIs. So it seems like the advanced AI would need to understand the human's most complex thoughts and emotions, and that's what neuroscientists are trying to do. So do you have any ambitions to tackle neuroscience beyond neuroengineering?
Well, I mean, I think we're going to make the input-output device and the software to interface with it, and I think probably, per the suggestion earlier, we'll try to open source as much as possible so people can take a look at it, and I think there will be a lot of others that build upon the work that we're doing. The same way that if you make a microprocessor or CPU or computer, that people will write lots of software that runs on that computer. But if you don't have the computer, the software is moot. So we're making the input-output device with the computer, and then I think probably there'll probably be a lot of other organizations, companies that build upon that foundation. So, yeah. I mean, one of the things that's I would sometimes wonder is that if you do have a whole brain interface and you can record memories, you're really getting into Black Mirror stuff here, but this could be one of them.
I also think it's worth mentioning an important point, which is that Neuralink didn't come out of nothing. There's decades and decades of research in the medical academic field that has really set the foundation for what is possible by putting these electrodes in parts of the brain and being able to read those signals, decode it for mapping it to some application. And being in academia before coming to Neuralink, I do think that there's a lot of opportunities for kind of the field to advance at a much rapid rate by having just better tools for observing the dynamics that are happening and then engaging with it in a seamless way. And I think it was Ian who sort of mentioned that it's almost as if like we're kind of building an oscilloscope for the brain, which I think is like kind of a beautiful analogy of just giving us a bit more abilities into peering into the dynamics and using those information, learn that to, I don't know, hopefully understand like what makes us and how the brain works and the whole shebang.
Hello. The presentation covered keyboard and handwriting-based input methods. How do you plan to develop an input model that will achieve much higher bandwidths for complex tasks in humans?
Yeah, this is a tough question, and we start exploring this with monkeys. As you saw, we have like we train many monkeys on very different tasks. It's still an open question that we're after. I think hopefully once we get to our first participant, it will be easier to investigate. One of the options we are exploring, as we showed, is to decode handwriting directly. This is a work that started at Stanford, and we are exploring here and trying to expand. There's also different, in addition to just decoding different things from the brain, we also try to provide the user different, maybe user-like interfaces. For example, we show different type of keyboards, maybe also swipe and other things that can help increase the communication rate. So we are kind of tackling those in 2 dimensions.
Yeah, just one other thing to add in that direction. As pointed out by many people here so far, this is a general I/O system that you can sort of plug and play in different places of the brain. There's other areas of the brain that can help increase bandwidth, for example, language or speech centers that can help you much more seamlessly communicate, for example, text, if that's your main thing that you're trying to do.
Yeah, I think just having this general input-output device will just so gigantically improve our understanding of the brain. It's hard to, the words can barely express. Like, right now we're just guessing a lot of what's going on in the brain, but if you have direct I/O, it's no more guessing. What we will learn about the brain with such a device in wide use is absolutely many orders of magnitude more than we currently understand. So I guess on that note, thank you for coming and thank you for watching online. And,